Hormesis and Aurelia orida. Aurelia orida is commonly known as the moon jelly. They are in the class Scyphozoa within phylum Cnidaria. Moon jellies are a cosmopolitan species living in the upper epipelagic zone. They're a common and easily recognized jelly. They're also widely studied by various countries due to their wide distribution across almost all warm and temperate coasts. The moon jelly is known for tolerating a wide range of environmental conditions and forming blooms across the globe. Research looking into how different conditions affect their life cycle has been thoroughly conducted, but what hasn't been thoroughly investigated is how pollutants affect them, specifically heavy metals. This is of interest to scientists because moon jellies often inhabit estuaries where heavy metal pollution is common. Recently, studies have been published that support the hormesis hypothesis applying to moon jellies. Hormesis is the phenomenon in which harmful substances are beneficial to living organisms when low concentrations are present. There have been 2 studies that have supported this hypothesis relating to moon jellies. A short-term 21-day study published in 2014 investigated the effects of copper and silver independently and in combination on moon jelly polyps. Higher concentrations of metals impacted the polyps more than lower concentrations of the same metal when used independently. When combined, they appeared to work antagonistically, with copper above a certain concentration reducing the damaging effects of silver. Interestingly, higher concentrations of the metals caused higher mortality and deformities, but it also increased budding and strobilation. The primary forms of asexual reproduction in moon jellies are budding and strobilation. Based on previous research, the polyps were not expected to strobilate because they were kept in constant conditions of temperature, light, and food availability. But they did, suggesting that it was most likely pollution stress induced. Except for the one treatment of high copper that caused 100% mortality, all of the treatments had a ratio of budding to mortality of greater than 1. These results indicate that these concentrations and combinations in marine waters may not be detrimental to polyps and may stimulate higher asexual reproduction, a hormetic response. Another short-term study just published in March of 2020 tested the independent and combined effects of mercury allowable by European regulation and hyposalinity on the asexual reproduction of moon jelly polyps. The mercury and hypersalinity did not negatively affect the polyp reproduction. Mercury alone increased polyp number and budding activity. Mercury with a salinity of 23 psu caused a 2-fold increase on polyp number and budding activity. These results support the hormesis hypothesis because asexual reproduction was stimulated when low levels of toxic agents were present. These results implied that sublethal contamination levels of estuarine waters could promote jellyfish proliferation, leading to more frequent blooms. The findings of these 2 studies opened so many more doors for research into how moon jellies affect the ecosystems they live in as dominant species. As heavy metal pollution increases in coastal and estuarine areas, it could cause enormous blooms of moon jellies. Many other species have reduced reproductive success and survival when exposed to these pollutants. A decline in competition for space and resources is likely to occur, allowing for moon jelly populations to further dominate ecosystems. Research on Aurelia orida over many decades has proven how hardy this species is and how they can adapt to so many environmental stressors. This new aspect of the hormesis hypothesis applying to this species has many implications that are sure to be further explored within this next decade.